Hello everyone. Have you ever heard of Pachyonychia Congenita? Welcome to your own channel ADP for Learning. Today we will explore a new topic, Pachyonychia Congenita, by addressing critical questions like what is Pachyonychia Congenita? Which gene is affected in Pachyonychia Congenita? What are the features or symptoms of Pachyonychia Congenita? And at last, how can we treat Pachyonychia Congenita? So without further delay, let's start with the first question: what is Pachyonychia Congenita? Pachyonychia Congenita, PC, is a rare genetic disorder that primarily affects the skin, nails, and mouth. It is caused by a mutation in any one of 4 genes that code for keratin proteins. Keratins are proteins that form tough fibers that strengthen skin and things that grow out of the skin, such as hair and fingernails. Although mutations in different keratins can cause many disorders, only mutations in keratins 6A, 6B, 16, and 17 are linked to Pachyonychia Congenita. This disorder does not affect lifespan, but patients do experience constant pain. Keratins are a family of related proteins with about 54 members. The keratin genes are located in 2 clusters, one cluster on chromosome 12 and another on chromosome 17. Now, with the use of simple animation, we will dig deep inside the topic of keratins and the Pachyonychia congenital disease that is associated with it. Starting with what are keratin mutations and disease associated with it. Keratin proteins are the main structural element of hair and fingernails. They are also vital to skin, where they add strength and durability. Keratins are a group of related proteins encoded by a family of 54 or so genes. These genes are located inside our cells, clustered together on 2 chromosomes. Different keratin genes are turned on in different types of cells. When a gene is turned on, it means that the protein it encodes is being made. Another way to say this is that the gene is being expressed. Keratin genes are turned on in pairs. Individual keratin proteins from 2 different genes link together. Then the partnered proteins assemble into tough rope-like filaments. In the skin, keratin filaments form a strong network of fibers that enable cells to withstand pressure and stretching. Occasionally, a random event causes a mutation in a keratin gene. The gene now codes for a slightly different protein. To understand how one defective gene can cause disease, we must first remember that everyone carries 2 copies of every gene, 1 copy from their mother, and 1 from their father. Here, 1 copy has a mutation and the other copy is normal. Remember, these keratin proteins must combine with their partner proteins from a second keratin gene in order to make filaments. There are 2 copies of the partner keratin gene, 1 copy from the mother, and 1 copy from the father. Now there are 4 active keratin genes in this cell. The protein products of these 4 genes form together into filaments. If just one of these genes codes for a defective keratin protein, the cell forms abnormal keratin filaments even when the other 3 genes produce normal proteins. The misshapen mutant keratin proteins do not pack together properly. After keratin's mutations, now we will go to the disorder of Pachyonychia Congenita. There are 2 different types of Pachyonychia Congenita. Mutants in keratins K6A or K16B cause PC1. Mutations in keratin K6B or K17 cause PC2. The keratin proteins produced from K6A and K16B are partners that link together to form keratin filaments. Keratin K6B and K17 are also partners. The 2 keratin gene pairs linked to PC1 and PC2 are active in many of the same places. People with PC show symptoms in the areas of the body where the mutant keratin genes are active. There are differences in the symptoms of PC1 and PC2, and variations in symptoms even among family members. However, the symptoms all come about in the same way. Wherever a mutated keratin gene is turned on, the cells form short, weak keratin filaments. Nails are thickened and weak. The cells of the palms and soles are so weak that they are ripped apart by normal pressure and stretching, causing painful blisters. In defense, the skin produces more cells, resulting in thick calluses. There are many other genetic disorders that involve other keratin genes. Keratin disorders have different symptoms, depending on which keratin genes are affected, and where in the body the affected genes are active. Now we will see the inheritance of Pachyonychia Congenita or PC. A mutated keratin gene can pass from parent to child in the father's sperm, or the mother's egg. Let's look at a case where a father with PC has a mutation in the keratin K6A gene. He has 1 normal copy of the keratin K6A gene, and 1 copy with a mutation. The mutated copy causes PC. His wife does not have PC. She has 2 normal copies of the keratin K6A gene. Each parent will pass 1 copy of K6A to each child. Which copy gets passed along is determined randomly, with each copy having a 50% chance of being passed down. Since both of the mother's genes are normal, she will pass a normal copy of K6A to each child. There is a 50% chance that the father will pass down a normal copy of K6A. In this case, the child will not have PC. There is also a 50% chance that the father will pass down a defective copy of K6A. In this case, the child will have PC. The chance of inheriting PC is the same for each child. This type of inheritance is called autosomal dominant. Autosomal because the gene is located on a chromosome that does not determine gender, making either sex equally likely to inherit the disease. Dominant because a single copy of the gene causes disease. So that leads to another important question. How do people get Pachyonychia congenita? Pachyonychia congenita has a predictable pattern of inheritance. People with PC have a 1 in 2 chance of passing the disease to their children each time they conceive. In other words, the disorder exhibits an autosomal dominant inheritance pattern. Children have to inherit just 1 copy of the mutated Keratin gene to exhibit symptoms. In some cases, a person with Pachyonychia congenita will have no family history of the disease. This most commonly happens when a mutation occurs very early in embryonic development, soon after fertilization. It can also occur when a healthy parent's egg or sperm cells acquire a mutation in one of the Keratin genes. The mutated gene is passed to the child, resulting in disease. In most cases, the affected child then has a 50% chance of passing PC to each of his or her children. As shown in the figure, normal keratin filaments form a strong structural network, left, that enables cells to withstand pressure and stretching. Mutant keratin proteins form clumps, right, resulting in weak cells that break open under pressure. That raises another critical question: what are the symptoms of Pachyonychia congenita? While every cell in the body has the genes that are involved in Pachyonychia congenita, symptoms only appear in places where the genes are active. Thick nails are the hallmark symptom of Pachyonychia congenita. In fact, Pachyonychia means thick nails. However, the most pronounced symptom for people with Pachyonychia congenita is painful blisters and thick calluses on the soles of the feet. Without proper keratin filaments, skin cells become fragile and cannot withstand pressure or stretching. Just walking across a room can put enough pressure on the soles of the feet to burst skin cells. Other common Pachyonychia congenita symptoms include blisters and calluses on the palms of the hands, white growth on the tongue, and a variety of cysts. Children often have different symptoms than adults. Symptoms more common in children include bumps around hair follicles, hoarseness, and intense, short-duration ear pain. Importantly, individual symptoms and their severity can vary between patients, even within a family. As shown in the figure, people with Pachyonychia congenita show a variety of symptoms, including, clockwise from top left, blisters and calluses on the feet and hands, thickened nails, cysts, bumps around hair follicles, and a thick white growth on the tongue. So that leads to another question: how do doctors diagnose Pachyonychia congenita? There are 2 forms of Pachyonychia congenita, type 1 and type 2. They are distinguished by slightly different symptoms and by which the keratin gene is mutated. To make a diagnosis, doctors typically look for a combination of Pachyonychia congenita symptoms. Because symptoms can vary, diagnosis is usually confirmed by genetic testing. Pachyonychia congenita is often misdiagnosed. The most common misdiagnosis for Pachyonychia congenita is onychomycosis, a fungal infection of the nails. Other common misdiagnoses are epidermolysis bullosa and palmoplantar keratodermas, 2 skin disorders that are similar to Pachyonychia congenita. So, at last, another question may come to your mind: how is Pachyonychia congenita treated? There is no cure for Pachyonychia congenita, and drug therapies are limited. Patients typically manage their own symptoms. Trimming and filing nails can prevent infections. Hands and feet can be soaked and rubbed to clean off blistered skin. Keeping tender skin moist and cold is often comforting. Special shoes, canes, or crutches can decrease pressure on the feet. Work is underway to provide better treatments for Pachyonychia congenita. A new technology involving gene silencing is providing hope for a cure. By inactivating the mutant keratin gene, researchers hope to restore keratin protein assembly and thereby strengthen skin cells. As shown in the figure, gene silencing is a promising new technology that may one day be used to treat Pachyonychia congenita. In one approach to gene silencing, the mutant keratin gene is turned off, 1, allowing the 3 remaining keratin genes to produce proteins, 2, that will assemble into proper filaments, 3. Scientists are also working on other gene silencing approaches. Thank you for joining me on this amazing journey of learning. We will meet again with new innovative topics. Meanwhile, if you have any doubt or feedback, please comment below.